For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 3, Episode 33, or coverage of the last days of ILC2022, starts now.
Today on Surfing the NASH Tsunami.
On this episode, we have a roundtable discussion with Mazen, Jörn Schattenberg and Ian Rowe on NITs and the evidence that came out on Saturday in discussions. We then have Robert Mitchell-Thane and myself looking at the aspects of patient perspectives that were covered during ILC 2022 and deep dive into what we may need to see next year.
Last week, roughly 5,000 hepatologists and hepatology stakeholders gathered at the Excel Centre in London, England, for ILC 2022, the first major liver meeting in 3 years with on-site attendees. Please join hepatology researcher and key opinion leader Professor Jörn Schattenberg and liver wellness advocate Louise Campbell, and today's guests, hepatology researchers and key opinion leaders Professor Ian Rowe and Dr. Mazen Noureddin, and patient advocate and PBC Foundation CEO Robert Mitchell-Thane, as they review some of the highlights and key issues of Day 4 of this year's conference, right now, on this special episode of Surfing the NASH Tsunami.
Today's ILC 2022 recap is sponsored by Dia Pharma. Dia Pharma is proud to support Surfing the NASH Tsunami in its activities to raise awareness of and foster discussions about the NASH epidemic. Dia Pharma offers non-invasive mechanistic biomarkers like CK18 that provide early biologically plausible indications of changes in disease activity for use in NASH drug development studies, for research use only in the US and Canada, and not for use in diagnostic procedures.
We got the usual suspects. We got Mazen, Noureddin, Ian Rowe, and Jörn Schattenberg, and myself, because Roger is in flight back to the US, just to unpick part of Day 4 of EASL in London. So, start with Ian. Give me your picks of the sessions so far, and I know you were moderating this morning.
The session so far, I mean, there's a lot to choose from, isn't there? I can't choose a session before saying what a treat it's been to finally get all together, to meet people who, I was sitting in a room with 3 people that before this week I'd not actually met in person before, and I think it's been terrific actually to get all together, meet people, have conversations that were not pre-arranged and on video. It's been really, really wonderful. But on top of that, I think we've seen some good science. And, we've had abstract sessions today about non-invasive biomarkers and also some data about new therapies or existing therapies in the case of semaglutide, but interesting data about tantalising for future outcomes. So it's been, another good day.
And Mazen? Your highlights?
Borrow most of it, actually. It's good to see people in person. You connect to them as humans. You can tell how nice they are as these guys.
You got to say that.
I mean, we all have. Yeah, keep it. We have. Our differences in sometimes interpreting the data, but it's good to be in person to discuss it, because it's just like the common goal that we want to get this better and improve the field and move forward. So that's what happened. As he mentioned, that we had exciting, and I want to congratulate EASL. Actually, they had a very good abstracts and program between NITs and outcome. The NITs performance. You have to follow what is out there for therapy. So we had a couple of exciting things about sema and cirrhotics, and the resmetirom safety data, I guess, if you want to call it, some efficacy in it. Let's just keep looking promising. So it's a good day, actually. It's a good NASH day today, Saturday. Yeah, and we still have more coming in the next hour or two. So
I might nip in with you on remotely to see what the feedback is on that, as we'll have to do this one short. You mentioned, obviously, having that discussion. I noted in the session this morning, that discussion with Rohit's presentation on the direct head-to-head with MEFIB, MAST and FAST.
Yeah.
Would that be an example of what you were talking about?
Yeah, indeed. So let me know when we need to talk about abstracts. I'm happy to talk about this. I think that was one of the highlights in the day, and we wanted to have scientific discussion, is like, what does head-to-head mean, and if we're comparing the right things, or which tests perform in which way with what we're looking for.
We'll talk about it now as the abstract.
Yeah, sure.
Because
there was a comparison today between multiple scores, included MEFIB, FAST, and MAST. FAST published in Lancet Gastrohepatology, MAST published in JHEP, and MEFIB published and got all of them within the last couple of years. And it's important to understand what was the primary outcome, which was more than 2 stages of fibrosis. I think MEFIB is an outstanding score that has showed persistently to be very accurate in predicting F2 and higher in NAFLD patients. And it did perform in terms of area under the curve better than the MAST and FAST from the presentation. But we had a friendly discussion after the session is about the purpose of FAST and MAST. And Laurent Castera actually also added to the applicability of MAST score, especially, we cannot go with MRI because of price and availability in Europe. Europe is a big part of this. So the FAST and MAST score are originally designed to look for at-risk NASH patients, NASH and F2 and higher. And when we put the MAST score, it was not rocket science, and I did not come up with anything novel at that time. It's like I looked at FAST, and FAST predicted NASH and F2 and higher, and I said PDFF is better than CAP, and MRE is better than stiffness. Let me just look at that. It was a question that I tried to answer. And when we came up with the paper, we said this is for clinical trials because a lot of phase 2A or 2B pre-screen or have MRI outcome in the 2As, and this is what we designed it for. So today, the comparison, as expected, the MEFIB had higher area under the curve in that cohort because it was mainly designed to look for fibrosis, and the outcome was F2 and higher. But I was also happy to see that when we had the MAST score, the PPV in that paper was I think in the 40-50%, which I think it's a good PPV. The PPV is a lot dependent on disease prevalence, outcome prevalence, and it performed well in Rowe's cohort, and actually was the Rowe cohorts. I didn't see the Japanese cohort in today's presentation, and it was in the 90%. So the overall, I think the comparison is, the message is we have multiple scores that trying to go beyond, might be controversial term, but the first generation of NITs, but they are the same machines. We're just trying to optimize to look what we're looking for. So from the FibroScan side, it's Agile 3, 4, and FAST, and each one is designed for one purpose. MAST is designed for another purpose, and MEFIB is designed for another purpose. And it was nice comparison. I think it was great presentation and just step forward in the field. I don't know what these guys think, but they're one of the smartest guys I know. So
you were moderating. Jörn was there.
Not sure I'm the smartest guy at this table here, but
I put him with you. So
you shared that session. I'll leave the last word to him on this. I think it was a great session, and good job on that, together with Jerome, obviously leading us through it. Mazen raises an important point. I'm just going to recapitulate a little bit on the concept as he highlighted. We have tests to detect advanced fibrosis because that's critically linked to outcome. That's important for patients. Okay, we want to identify the patients with advanced fibrosis. And MRT-based technology is superior to some ultrasound-based technology, superior to just blood-based technologies. I think that's a common theme, and the refinement of these scores has been elegantly done by a number of key players, Rohit, Mazen, and that's nice. And the other set of tests is really for trial enrichment, where we're looking for activity and fibrosis. So there, all of a sudden, we combine two aspects of the disease. Activity is always a little bit difficult to be assessed. The biopsy is easy missing activity. And one of the comments today that came up to the Jenny Lee's paper, the LITMUS Consortium tried to employ machine learning algorithm, was actually, well, how sure are you that you're looking at the right condition just looking at liver biopsy? And are you missing a balloon cell in a slide that histology is red, and maybe the case is counted as non-NASH, where really it is NASH, and it revisits the whole, problem of the field of what's the reference standard. And I agree with Mazen. We've seen a lot of data emerging linking NITs to outcomes. So I think we're even one step closer, and I'm going to use this term here, to move beyond the biopsy. Mm-hmm. And
Donna Cryer would be clapping.
I think that's a fast track, and I agree. EASL has pulled back together a great set of speakers, attendees. I think it was over 5,000 in person this year, and online followers. So I think we're in a good way.
Ian?
I think that is a really excellent summary of the session. One of the things that I think, we've got lots of good tools now. Are they perfect? No. Are they close to, being as good as we're going to get? Well, maybe they are. Perhaps the next step is really to try and coalesce behind, one or two or three of them, depending on the context of use, whether it's use in primary care, whether it's about hepatology practice or enrichment for clinical trials, but to really say that these are the ones that we're going to use, because standardizing would help, mean that everybody was talking a common language. And then we could think more and more about clinical utility, cost effectiveness, and exactly how we're going to use use them in practice, because that's the next step. Research is great, and I think the work that's been done by lots of investigators, but particularly also by LITMUS and by NIMBLE, taking us to that point where we can get behind one or two of these biomarkers.
When I was watching those sessions this morning, and you alluded to about accurate tests, I go around the posters and I've read through a lot of the abstracts. I don't know which version of FibroScan is being used. I don't know whether it's first generation, doesn't have cap, or it's the old cap, or it's continuous cap. I don't know whether the IQR is less than 30% and in the high range. I don't know whether it's less than 10%. Is it time to nail down literally how accurate FibroScan is and show that on these slides that the average IQR percentage was less than 10 in this population? Because that would be way more accurate than the average population in this trial was 30%. So really drilling it down, because we know smart exam and continuous cap are way more accurate, which should make FAST score more accurate and ALL 3 and 4. And we see it for standard deviation and lots of other things, but I've not seen it in any posters. And we're now on third generation FibroScan. Is it time to actually include that data?
So I think that's a really important point. So for laboratory tests, there is a quality control, quality assurance program. For imaging tests, there's QC for your machine, but there isn't necessarily QC and QA for everything else. But for point-of-care diagnostics like FibroScan, then it's a bit less clear what the quality control and quality assurance is. And I think it would be, how much is that the technology, how much is it the operator, how much is it your patient population? All of that is going to play into how we interpret those findings, both in the research and trial settings, but also in the real world. I guess the adoption of new technology is, in some circumstances, the pathway is very clear, but in some of these point-of-care tests, it's perhaps a little bit less clear. And as I said a bit before, we have to be talking a common language. Everybody needs to know that a liver stiffness of 12 in Leeds is the same as a liver stiffness of 12 in Mainz or in Houston. Yeah.
Very well said. And since we're on a podcast that is attended by a lot of people, my colleagues and I, we do abstracts reviews and we edit journals and we review papers. And when you review an abstract, and if you would have the IQR and what was used and the quality control and all this abstract is more likely to make it oral than a poster. So I think we should emphasize on the message that there should be consistent reporting in FibroScan measurements. And if you present your science in a more clear way, and that way you don't leave these questions in our minds, like what happened, what if it's this, what if it's that, you will more likely to have it in a better journal, a better form of presentation. So it's call for action on this podcast. Please report it. And but especially we're now switching generations between the machines and all that. So
and I think
very good point.
Stephen made the point on Wednesday morning about trial enrichment. If you have a FibroScan in a small trial, phase 1 or phase 2, where you've got very small patients, but your average IQR is about 25%, they're not going to make that through, and they're going to drop out of the trial very quickly. So we should be, I suppose, setting the target we want to try. And in smaller numbers, it's very difficult. But I think it's not anything I've ever seen reported. Yet we all use it very well. And I look at what IQR we get, and I look at the standard deviation. I try to get less than 10 on both sides to get it as accurate as possible.
I can tell you, actually, I want to give credit to Quentin Enstey that the MAST paper, I think he was the handling editor, and we missed a couple of, I don't remember, was it the IQR or something, or he was very systemic in terms of how we should report the FibroScan, and they improved the paper. Editorial board and JHEP really pushed us on the MAST paper in a very good educative way, and they had quality there and made sure everything is consistent in terms of how we're reporting things. And JHEP, as you know now, is the top journal in the field. So it's required by top journals now to be consistent in your description. Which probe you used, now like we have the cap, the smart cap, and the IQR and all this, and why you use the FibroScan and which cohort.
And I think going back to your point, people say, oh, well, it's a little bit flexible and it's this and it's that. And addressing that could potentially help, because you can look at your data. John?
Yeah, I echo that. I think, we're seeing lots of data emerge, and to look at that quality aspect is crucial. I'd like to change sessions for a second and think one more further. Parallel to our session we had this morning with the NITs, there was a public health session on NAFLD. Now, unfortunately, I could not attend in person, but a colleague of mine presented a screening study, the SEAL study, which is actually now published in JHEP and the actual current version. So this addresses the referral pathways in screening and primary care. Something they did is to identify patients using non-invasive tests. Now, in the SEAL study, we used APRI, something we'd probably not use from a today's perspective, but it's an at-risk at-risk population, and then we try to refer patients. And it was a very good study, systematically, prospectively done. Just to mention one fact is that over half of the patients that were at risk using APRI did not show up with their secondary care appointment. So I think we're improving our metrics, we're improving quality, but we're losing patients on the way. Revisiting the concept of models of care and how do I link someone with a diagnosed liver disease, or how do I make the patient or the physician aware that there is additional treatment, or in the first step, diagnostics recommended, is something I think we have to think about and address in the future.
Oh, thanks for bringing this up. We usually miss these sessions because we want to be in our sessions, which actually I'm going to flip it back. And to you, I think you did a great job today and brilliant presentation. I'm not trying to take your role now, but I think it's important to come on the podcast if you want to give it a minute or two. I think that was brilliantly done, and you presented in a very unbiased way in the right patient setting. So I'll stop here. I'll let you comment on it.
Well, thanks for putting that ball on the penalty spot. I'm going to take a shot.
We're getting him into football.
I'm Roger.
We'll get you into football, Mazen, for the World Cup.
So this was screening data of the NASH trial, which is an ongoing phase 3 trial exploring resmetirom in patients with at-risk NASH, defined as NAS4 and higher in F2. And they looked at how well did the FIB4 perform in identifying these patients. And just to recap on a number of the central findings for me, 37% of all patients are qualified based on liver biopsy to be enrolled in a clinical trial had normal ALT. I think I said that in the presentation. ALT is still the test that triggers referral to my clinic in everyday life, not a FIB4, not an APRI or sometimes abnormal ultrasound, but the ALT. And I thought it was interesting to see that 1/3 of those patients in a clinical trial, where we think they benefit from experimental therapy, normal ALT, and 80% had below 2 times upper limit of normal. And I know enough colleagues, no blame, they'd say, well, it's a mildly elevated ALT, come back or, stop doing. I think we're at a point where we really have to talk about what's normal and how we can educate, peers, colleagues, and the field. And the second, just to revisit the abstract, so the second finding was FIB4 of using a cutoff of 1.3, and actually in the same session, Jérôme Bossier gave a great talk addressing the same aspect in the type 2 diabetes population is not the best test, and it's going to miss consecutive numbers of patients with at-risk NASH defined by NAS4 and more in F2. That was my key take-home. I don't know if you have any follow-up or questions to that. And again, I'd also like to loop in Jérôme Bossier's talk here on the type 2 diabetes FIB4.
You hit 2 birds with one stone.
I think it came up in the afternoon because I was talking to you earlier, Rohit Lumber's session, looking at diagnosed cirrhosis in a treated population and an untreated population using ISHAC. Not one of the average FIB4, and everybody bar one patient was in 5 or 6, and that was in the non-treated, the placebo arm, had a FIB4 of 2 and 2.2. So those were full-blown diagnosed cirrhotics, and they didn't hit the marker for FIB4.
And not to take away from FIB4, I think Ian said it earlier, it's the context of use, the right population, and disease prevalence. So it varies between primary care versus tertiary care, diabetics and non-diabetics. So we should be just careful with what we're using it for. And even cutoffs, like with the MAST and FAST and all this, it's them. I don't want to go too deep, but the cutoffs will depends on the disease prevalence. And which was 18% for NASH and F2, which is regular hepatology practice. If you go on clinical trials, it's about 40 to 50% NASH and F2 because of the pre-screening. So if you really, there are collaboration kind of curves in that was done in the FAST paper, and we tried to do it as well, that maybe there will be different cutoffs, and I don't want to confuse people. But back to the context of use and the right population, there are differences, and it's important to say it's not, we need to compare apple to apple, the right test to the right test, the right population to the right population. So
and I think what I took from it was a good test in a primary care setting where you want to start to enrich your population, but not a great test in an enriched population. Would that be right?
Yeah, I agree. And just to reflect on Mazen's comment, I think the field is complex and we're seeing a lot of tests and there will be a lot of refinement. I think the way forward is to use some diagnostic tools tools and maybe automated systems that do have all that in the back end, and the physician that knows I'm operating in this system, this is my patient, will get an easy answer through an automated system, maybe a lab system, maybe the electronic healthcare system. The physician, even the normal hepatologist, they will not have to have a deep understanding of these complex algorithms, just the cutoffs, and they can be put in a computer system or an AI algorithm. And I think that's the way forward. We got to automate some of these things.
Just to follow on from that a bit, one of the values of some of the scores, and whether it's elastography-based scores or even FIB4, is that they're continuous. And Rohit Lumba made a really interesting comment, I like simplicity, I want to draw cut points. But the reality is that the cut point is just an artificial divide that separates a probability of F2 or less from F3, F4. But again, that's just another ordinal surrogate for outcome. And we should embrace some of that complexity and think about likelihood of disease. So when you're automating, you could define your thresholds and say, well, as a primary care practitioner, what I want is to have a group of patients identified who've got a 10% risk of outcome A in 10 years. And that's what the cardiologists have been doing for decades now. And I think as we see more data emerging for the longitudinal outcomes associated with biomarkers, then we'll be in a much better position to do that, rather than only, and it's not only, it's been a massive endeavour, but only looking at biomarkers with relation to fibrosis stage. And this is a long journey, and the train's only just left the station, but we should think about what the destination is so that we're sure we're on the right train. I think I've probably stretched that analogy too far.
I totally agree. Plus, to add to that quickly, clinical trials are targeting certain type of population, which is the NASH NF2. And if I want to move away from the biopsy, I want to know which one is NASH NF2, that my drug will be as close as to biopsy. Again, it's not the best gold standard, but that's how we're doing clinical trial. So back to the context of use.
From my perspective, there's been a lot of data and a lot of abstracts shown about the predictive outcomes and liver-related outcomes and cardiac-related outcomes in a lot of the non-invasive, which is nice to see. We're seeing more and more data. Do you think that will come forward to the FDA, things like that?
Oh, 100%.
Yeah.
That's what they're looking for, indeed, and that's what they're going to replace the liver biopsy. They told us, the example that came out from one of their colleagues in the FDA is, what I want you to show me for NITs to replace liver biopsy in registry trials is the following. In the HIV world, and he gave the example of HIV, we had to have a drop in viral load correlating with outcomes before viral load as an outcome. Now, rather, if we agree or disagree, and if it's stealth or not, that's what they're looking for, or that was one of the comments that were made. So outcomes is very important, and thus every time people come up with a score, I'll give you an example. We looked at the MAST score in terms of predicting outcomes, and we have data that is coming out soon that it does predict, and in each zone, and the highest zone is significant hazard ratio. The intermediate zone had a little bit hazard ratio, but not as even close to the higher one. We looked at it at FAST. We looked at Agile 3 and 4. It's connecting to outcome is very important in a way, but again, the context of use is a big topic, and it has many categories. Outcome is sexy, if you want to say, and the regulators approve a biomarker in way or another. So everyone is looking for outcomes.
I think it's also important in the discussions with the colleagues, the ones are not so deeply involved in the field as Mazen, Ian are in the discussion. We got to show that this has an impact for patients. Indeed. And it's a burden for the field, but it's important we carry this forward. We have to remember that the number of events, cardiovascular events in a NASH trial is not going to lead to a significant difference between treatment arms, no matter how effective the drug is, because A, we don't enroll patients with the highest cardiovascular risk, and B, the numbers in the NASH trials do not match the ones in the cardiovascular outcomes studies. As a matter of fact, those patients are excluded because they are in a high-risk category. So that's one. But here comes the strength of consortia or initiatives. Mazen has just started up one. We've touched it briefly here, which is NAIL-NIT. There's NIMBLE, there's LITMUS. There's another big study cohort in San Diego. And I think they are going to be collectively support that outcome at the linkage to an NIT. And I think here the world of academia is really supporting and answering these questions.
And we've been joined by Robert Mitchell-Thane from Global Liver Institute and PPC Foundation.
Hello.
From the patient's aspect, what have you seen at the conference? Is there enough? Is there enough attention? Is it in the abstracts enough?
For patients?
Patients and related outcomes.
Good question. I think through the COVID experience, the AASLD put together a really holistic, complete patient integration program, which I think is set as the standard for where we need to go in the future. Having been here for a physical meeting now, it would be great to take the lessons learned from the COVID era and maybe incorporate more patient involvement in terms of symposia, in terms of guiding some of the less experienced physicians, and also allowing patients to share patient-reported outcomes, experience measures, those kind of things to, again, help inform the discussion and the way forward.
Have you seen anything that, I suppose, rocks your world and you think that's really going to change what's going to happen to patients in the next year, 18 months?
Again, another good question. So I've seen things that have rocked my world in the sense that I hope they change. So, for example, when you look at conversations around FIB4 and the number of patients that are actually being missed through that mechanism, I have concerns, and I hope that there will be some answers that come forward in terms of making sure that every single patient is possibly caught at a much earlier point, particularly when you're looking at self-management, when you're looking at the emotional, the psychological parts of the self-care that goes with it. The earlier we intervene, the more chance the patient is going to have of taking a much more direct and proactive approach that's going to be successful.
And with the FIB4, we were talking about the FIB4 earlier, in the fact that it's more or less a good test to enrich a population from that and do the sessions like that. But yes, it had weaknesses. What is the biggest thing you've noticed between, you've been to EASL before, haven't you? Between the previous ones and this one, apart from a smile on everybody's face, because I have noticed that outright.
No, absolutely. The elation of seeing people in the same room. But I think with the elation comes the opportunity to have a much more robust and frank conversation. So in terms of bad practice, if you and I were to have a big long pause between your question and my answer, that would be exactly what we're used to in Zoom. Because you come in with a point, but you don't want to talk over the other person, and you're 1,000 miles away, so there's a slight delay, and so you have these big pregnant pauses. A bit like that, which don't make for great interesting listening. However, when you're all in the room together, somebody says something and you can spark off. Yeah. And you're like, that's a great point, let me come in here, and then boom, and then suddenly the conversation just moved miles and miles down the road because you're sparking off each other in a much more meaningful way. That has felt absolutely amazing. And when you have such great minds coming together to actually focus on what they do and what they do well, that spark, I think, has moved a number of conversations on.
And it's been reassuring to see a lot of the data that's been produced, topics and so diverse. Yeah. And I know you've done a lot of the rare diseases while you're here.
Yeah, absolutely.
But you also gave a session on stigma.
Yes.
In fatty liver disease and NAFLD.
Yeah.
Is that something we take seriously when we design trials and patient outcome reports? Because, for example, somebody who's a cirrhotic and maybe uremic, I think around about 60 to 70% of men who have uremia can't get an erection. That affects their quality of life.
Of course.
But I don't know that there's really much literature. I did a literature search here for anything with menopause. There was some at postmenopausal, but it was only in hepatitis C. There was nothing on sexual dysfunction or something that really, really affects people.
Absolutely. So hopefully it's not the fact we're in London and we just don't talk about sex here in the UK. So funnily enough, when you asked the question earlier about EASL and things that we did well here for the patients, the patient forum was really well attended. I think it was a really helpful session. And Frank, now I can't remember his full name and we can look at it, but Frank spoke on behalf of EASL. He's an Irish doctor and he was amazing because in a very empathic, natural and genuinely meaningful way, he said, we're listening, educate us. We want to come together to genuinely tackle stigma, which was wonderful. And there was a buzz in the room when he was speaking because he meant it. And that's great to see. Stigma is an interesting topic because you have external stigma. So, if somebody has fatty liver disease, then there's the criticism and there's fat or there's judgmental or you've done it yourself and all of those aspects. But there's also internal stigma and whether you choose to actually talk about what you face inside. So there's a huge gap in the data set, but I wonder if we are part of the problem. And I think it would be helpful if we became part of the solution. Now, that said, as a middle-aged man, if I were to have sexual dysfunction, I would be particularly, spectacularly choosy about the people with whom I would share that. And part of that is the difference between the sexes. So, for example, my wife were to go to a girlfriend and say, do I look fat in this? The friend would say, no, you look amazing, you look beautiful, and I love you for who you are. If I were to go to my best pal and say, am I looking fat? He'd be like, you know what? I know 5 fat people and you're 4 of them. And it's a totally different support structure. But sometimes there are topics that are too sensitive to have that particular type of fun banter support structure. But if the doctors are going to talk about it, if the doctors are going to address it, if the community is going to address it, we have to do that first. And we have to lead from the front and say, you know what, this is an issue for me. And it might start with 1s and 2s, but from that trickle will come the numbers required to get us talking about the topic. And then once we're talking about it honestly, more numbers will come. And once the more numbers come, we can have a much more bigger data set, which then not only acknowledges the issue, but helps us address it.
But I think you're right there. The solution does have to come from us as practitioners, nurses, doctors. Everybody should be having that conversation the first time any patient's educated with liver disease, that you might find that you get sexual dysfunction. You might have an altered body image. Let's talk about it now, because it breaks down that. And I think it was Jeff Lazarus in the consensus report that highlighted institutional stigma within hospitals. Not so much because it's designed, it's because most hospitals don't have a liver speciality nurse. You can go into any trust in the UK, and I suspect in the US or France or Germany, and get a cardiac specialist nurse or a diabetes specialist nurse. For something that is coming through very much as part of the increasing population of hospitals, we do not have that person. And that affects the quality of referrals, the length of stay, the information that a patient gets, and a lot of people don't recognise encephalopathy. They then put somebody who's agitated and irritable on morphine. That's the last you ever heard of them. We do need to address that in any way, and that should be in these pathways. It should be part of that conversation. It should also be in clinical trials, because it should be an outcome measure to measure it, because then it becomes in the conversation. And I think quality of life from all of that is absolutely key. And I've seen plenty of patient-reported outcomes. In fact, Zabir Younossi was discussing it yesterday, and he is by far one of the best experts on patient-related outcomes from taking the measurements and analysing the data.
Absolutely. So I think you make a great point. This is the clinical experience for the patient. What's important? So when you talk to what's important about doctors, they'll say your blood tests, your prognosis, and those kind of aspects. What's important to patients is quality of life and how they feel inside and out. What's important to nurses is that middle ground where the biochemistry and all the tests impact upon quality of life. And nurses have a much, much more conversational exchange with patients. And I think this is the key. We're going out all around the world, and to some extent, in every single country, there's still doctors on a pedestal. I mean, thankfully, we're past the days where we bow and curtsy as we come in and go out, but there's still a huge amount of that kind of psychology that goes on. He's a doctor, he said that, it must be right. And so when you're living in those circumstances, you can't necessarily have an open and honest conversation about your deepest, darkest fears, and this is how it affects me, and this is how it affects my life in private. Whereas when nurses are much more conversational, much more informal, I think it helps enormously. And so yes, there are aspects to this that are important to come from the clinical part, but we as patients need to meet halfway, and we need to be brave, we need to be bold, we need to be honest, because frankly it's not going to fix itself. So let's take that first step.
I think you touched on a really good point there, because the minute you tell somebody they're a patient or diagnose something, they hand over responsibility. What are you going to do for me, doctor? Whereas if you don't ever make them a patient, poor liver health it could be detected without making somebody a patient, because you can address it in lifestyle and things like that for some extent. But there is a whole host of evidence the minute we paternalise the whole thing, and that happens in clinical trials. I used to do a lot of hep C treatments, and we based our whole treatment around a clinical trial pathway. That was how they got good success rates. So therefore, when you bring it out of a clinical trial, you have to do the same thing to get that success rate. And one of my concerns is that when we do all of these clinical trials for NAFLD and NASH, once we get into the real world with drugs like resmetirom and things like that are likely to be approved in the next few years, and pamidine, I think it is, that we've heard a lot about this conference is that if you don't deliver it in the same way, you won't get the same outcomes.
Okay.
And those outcomes will fall. And then people feel, well, the drug didn't do it and it's not doing what it's supposed to do. We have to pay very, very close attention as an industry, as a clinical research program, that when it rolls off the production line, that we've thought of everything. How do we maximize that? How do we, in hepatitis C, we did right drug, right patient, right time. And if it was never the right time for you, then we weren't worried. I've had people wait 10 years to get into treatment, but they kept engaging.
Absolutely.
Or people who've disengaged and then come back. And I think that hits to your point that we have that conversation, but we do get more time. And I think when you look at placebo lines and that for trial, it's often the nurse's engagement. And there's been some evidence here about that engagement in a couple of posters and abstracts on just one engagement with FibroScan with a nurse.
Yeah.
Or there was a very good session in the nurses and associates yesterday that showed that even 5 years down the line, there were certain people who would reduce their cap, and they were only focused on the cap. So it means something, but it also means something, that engagement.
No, absolutely right. So I'm going to take a slight step to the left because this is a personal button of mine. Now, so I've been involved as a patient advocate for 20 years, and I've been diagnosed with fatty liver disease for 5. So I'm coming from two entirely different perspectives. But I would ask each and every single person who is affected by a condition to not only describe themselves as a patient, but to absolutely own it. So when you come to conferences like these, you have clinicians who are clinical experts, but you also have patients who are patient experts. And so when you see a clinician, it's a 15-minute appointment, and maybe that's once a year, once every 6 months, and that's a tiny, tiny snapshot of your life. But when it comes to living your life, when it comes to dealing with the condition, physically, emotionally, psychologically, you are absolutely the expert. So please, anyone who watches, anyone who's listening, please take ownership of the word patient and become the patient expert, become part of the solution. Because when you look around at these kind of conferences, the clinical world need us to engage to help find the solutions. And the sooner we do that, the sooner we take ownership, and the sooner we realise the power we have with that expertise, I think we're going to help the whole process.
It would be interesting post-conference. I'd like to see the British Liver Trust's data, Echosense's data, and eScopic's data, because they've all been scanning people. And I have certainly seen a lot of soft fatty livers in our own community of physicians and nurses. And I think that surprised some people, but it'll be interesting to get the figures, because I know when the British Liver Trust went into the Houses of Parliament at Westminster, about 30% of all MPs they scanned needed a referral to their GP. Now, that might be for stiffness, that might be for CAP. They weren't using it before, but I do believe they're using it now. So it will be very interesting if we can get that data. There's also sometimes difficulty when patients say, you're telling me to do this, but you don't follow it yourself. We're all human. None of us are perfect, and we may eat more, drink more at various different times. It's a very hard conversation to have. People say to me, because I'm relatively slim, you can't talk about obesity or diet.
Yeah.
No, I can't talk about how it feels to have that, but I can talk and listen. And I can empathise with you, and I can give you suggestions on what you tell me that might help.
Yeah.
We shouldn't presume to be able to just prescribe and expect them to do it and say they're not following my rules and regulations. They're just difficult to engage. But you stay with somebody, they will engage. It's when you disengage as healthcare that we tend to run into a problem. So when we lose, Stephen Harrison did a very good presentation this afternoon on the resmetirom data from Maestro. There was a non-cirrhotic population. I think it was F2. In fact, the FibroScan had to be above 5.5 and less than 7.5, and a cap had to be over 280. So it's just soft fatty livers. But because it was during COVID, they did lose a number of patients to follow up and people who didn't want to come back. Quite rightly. I think a lot of patients didn't take at least 2 months worth of medication. I think there was a hiatus in it, but it still showed benefits. It still met its endpoint. It still showed that the side effects were tolerable. That sort of data is really important, because that's the sort of data we talk to people about trials. It might not be the trial you go on, But actually, we've got to engage people.
No, it's a fair point. And so I jumped back to my day of diagnosis when we were having this conversation. And I remember, so you'll remember the data that actually comes back, but I remember being told at the time that a 10 to 15% reduction in my weight has an enormous implication in terms of reducing my risk. You might remember the percentage off the top of your head. I certainly don't, but it is monumental compared to that 10 to 15% of weight.
Yep.
And so even at that moment in time, I thought, 10%? I can do that. And just having that focus was enormously helpful. And going back to the clinical appointments and everything else that goes on, so there have been a huge amount of data and studies that tell us that from the moment you're spoken to or with in the clinical appointment to when you close the door when you leave, you lose somewhere between 40 and 70% of the information that was transferred.
Yep.
Just straight in, straight out. And the other part, so coming from my professional side now, when I look at emotional, psychological, and physical self-management, is that when you perceive a threat, you start to operate in the emotional part of your brain. So you're not thinking solutions, you're not thinking win-win, you're thinking, I've heard a threat, and so I'm going to jump to survival mode. So you're talking flight, you're talking about adrenalizing the body, all of these things, none of which help you in the process. And even the simple conversation of somebody saying, you know what, you could do with losing a few pounds, is enough to trigger the emotional part of the brain, which then creates this defense mechanism response. And so we as human beings, instead of hearing that, and frankly, someone could say to me today, I can do with losing a few pounds, and it's absolutely 100% true. But when you hear it in such a way that you then react emotionally, when the defense mechanism comes with it, you're then again becoming your own barrier to the solution. And again, I think it's important to acknowledge that, because if that happens to you, that's okay, because that's a perfectly natural response. But once you're aware of that, you've then got the chance to come back a couple of days later and think about it in a much more level-headed, cognizant way to say, you know what, I can do that 10%. Now, it's taken me years to gain that 10%, so it's going to take me time to lose it. But actually, 10% I can do.
And I think what we know, and there's been, again, abstracts here and abstracts earlier on in the year, we've discussed it on the podcast, the quality of the diet helps. And so, for some people, they won't need to lose 10%. But if we're not measuring that, it's just a carte blanche.
Yeah.
There was a very good session today on type 2 diabetes and its relation to cancers and that in a session. And I have to say, it was delayed because she had no reception in Finland. She drove her car to the top of a hill to get reception, and most people stuck out the lecture with the intermittent. So Helena, and I can't say her name, but she deserves the award for gone the furthest mile to get her session done. And Helen Reeves stepped in with the cancer risks.
Okay.
And fatty liver, in any of the studies and the meta-analysis, increases the risk of primary HCC. We know that, but again, it's nice to see the data. There was a Swedish study that didn't suggest that you screen. The trouble is the WHO is not meeting its SDGs on cancer. So maybe we have to revisit that somewhere along the line.
When you say SDG?
Sustainable Development Goals. The one they're definitely not going to hit is locating and the rise of cancer. And it's predominantly because the rise of liver cancer is so exponential that nobody can keep a track on it. NHS England have tasked most areas of the UK to do 1,000 fibro scans to locate higher risk patients. Whether they get that done, because we know that we don't have uniform access to FibroScan. I think 74% of the country don't have it. So we talk a good game, but as Jeff Lesler has said, the UK is top of the list of somebody who can get a NAFLD program in, but we all failed. We just failed less than somebody else.
No, that's a fair point.
What I commented on yesterday's podcast was what I now want to see is not next year redoing the same data in a different way. I would like to see something happen with that data. We know that primary care, where it works by scanning or testing people in their community areas, filters out the right patients to go to hepatology. And we've known that for a long time, but we don't put it into any pathways yet. It's just funded by those local areas like Nottingham, Hampshire, Southampton are the three that I can think of off the top of my head. And that must be really frustrating. From a patient network perspective, we talk about equity, equality, diversity, and it doesn't hit any of those.
So funnily enough, thinking about your Finnish presenter who went that extra mile, and when you think about the number of clinicians that we know personally, that we've met, and some that we've heard in these podcasts, these are clinicians that will go the extra mile, and yet they're hitting systemic blockages.
Yeah.
And I think it's important, again, for patients to know that sometimes the frustration that we feel is being felt by our clinicians, and they're trying to do their utmost to help us. And sometimes it isn't the clinician who's the barrier, the obstacle. And I think it's an important realisation.
Her name was Henele Yiki Jarvinen, and I apologise now for my Finnish pronunciation, but she was doing NASH and type 2 diabetes, and she came up with lots of medications, where they work and where they don't work, where they help weight loss, where they don't help weight loss. And the over 10% was able to reverse NASH. So there's a lot of that evidence, but there's also been lots of evidence about the quality of diet can actually change your liver fat quite substantially. In a year's time, coming back, what would you like to see different? What do you think will be on the agenda? If you had a crystal ball, and what would you like to see on the patient agenda from clinical trials or from lifestyle?
I would love to see patient-reported experience or outcome measures. So patients talking about, actually, I transformed my life. Patients talking about the impact that support groups have had to help them manage that change. And managing change is a multi-million dollar, pound, yen industry. And nobody's found the one answer. But I think if the patients can come together and say, this is what we found is helpful, and be part of that conversation, be invited by bodies like EASL, by the clinicians, that I think we'll get that ball rolling just a little bit faster. And that's the biggest single thing I would like to see next year. In terms of what we will see, I honestly don't know. I'd be curious to see what happens. I think AASLD will be a good guidance in terms of what the trial results are. The one thing I do know we will see will be the fact that by this time next year, Arsenal will have qualified for the Champions League. And Spurs will be
Dream on. Roger will not be happy. There's a reason he doesn't want you on this podcast. Yeah, no, Roger does. But yes, and Tottenham will have qualified to... No, it's great. And I think this movement forward is absolutely vital. And I think the dialogue that's happening is now happening across the board.
It is.
And I certainly think people are more receptive to looking at the other facets of a clinical trial rather than just drug development. And I think there are obviously some big companies that sort of straddle obesity in children as well as diabetes, as well as fatty liver, Novo Nordisk for one. What I do think next year, it'll be interesting, because I was in San Francisco when Ed Gain delivered the results for sofosbuvir for the first time.
Yeah, wow.
And every room, an outside room, absolutely rammed for that data. Will we get that next year from the resmetirom studies or something like that? I don't know. I'd like to think so. But if not at EASL, ARSL maybe next year.
I would think so. So I remember the olden days coming from the PBC world, where the Hep C lectures would be mobbed, and it's like, now we're going to talk about PBC, and empty, and then we talk about NASH, and you get one or two interested people, and that's absolutely changed. And I think there are more great minds. I think there are more meaningful networks, and I think there are more companies than ever who are looking at this in a serious, joined-up way. And I hope that this one is the one that makes a difference.
Can I ask you, PBC is often ruled out in any NAFLD and NASH study. So is there any research going on in PBC patients with NAFLD and NASH?
That's a great question.
Because when I fibroscan people, I then have to have that dialogue about diet, because it's two conditions. And I think it's something we need to be aware. But is there, I presume, like hepatitis C and HIV, it progresses quicker, hepatitis C and hepatitis B, the more you have, the quicker the progression. But I haven't seen much evidence on it.
No, that's a great question. So I'm not aware of any holistic study in terms of looking at the two conditions together. And you made me smile, because so at the BBC Foundation, they've worked really hard with the Global Liver Institute, particularly on IND, to raise patients' awareness that just because you have PBC, and it's not just a PBC thing, PSC, AIH, you can have a whole number of other liver conditions, and that will never stop you developing fatty liver or NASH, because they're two entirely different mechanisms. And, for all the reasons we know it can affect the general population, it can also affect the liver population in the same way. And so we've worked really hard to raise patients' awareness that, okay, yes, I may have PBC, but I also need to look at this aspect of my life as well. And when you look at features such as the fatigue in PBC, the number one way to overcome that fatigue is meaningful activity. And I'm really careful not to say exercise, because when we say meaningful activity, singing, pushing the kids in a swing, all of these kind of things can, and you don't need to wear lycra to do any of those things.
positively by benefit.
But those are really, really important. And so there is much more awareness within the patient community. But no, and you raise a fantastic point. I think it would be really good to see some of these comorbidity types.
Even just as small studies, because otherwise our patients with other liver diseases who have fatty liver as well will be exempt from any of these medications and they'll be treated off-label. And we don't know what the effect is. No. I haven't seen a lot of data on it, or whether or not we're looking at it. That may just be because I haven't searched the database for the trials at the moment. But
so, no, to my shame, I'm not aware of anything either.
So maybe it's an unmet need, rare liver disease that also has fatty liver, because we spend a lot of time talking about maximising the one disease that's genetic and will progress for most people.
Yeah.
Let's keep it as healthy as possible.
Absolutely.
Because there's a lot of focus at the moment on NAFLD. It's moved from hepatitis C to NAFLD. Yeah. And I'm very aware that rare liver diseases are always at the bottom of the pile because there's not a lot we can do currently for them.
We're the poor cousins. We know.
Well, thank you for your time.
It's been an absolute pleasure. I was just going to say, it's when I listen to the podcast, I think, well, that's a cracking question. Well, there's another cracking question. It's really great to hear because you spark debate. But I can tell you, sitting here at the receiving end of those great questions, it feels entirely different. But it's been a pleasure here today.
It's my eyes piercing in there. And I think we lost Jörn and Mazen and Ian to the parallel sessions that were going on. So I'd like to thank them for their time. I'd like to thank you for your time. And hopefully this time next year or at ARSLD, we'll be discussing more data and potentially any trials in rare liver diseases that also have fatty liver so that we can try and maximise the outcomes. So thank you, and hopefully I've stepped in for Roger to the breach, dear friends, and we can get this out for everybody. But thank you.
Pleasure.
Today's ILC 2022 recap has been sponsored by Diapharma. Diapharma is proud to support surfing the NASH tsunami in its activities to raise awareness of and foster discussions about the NASH epidemic. Diapharma offers non-invasive mechanistic biomarkers like CK18 that provide early biologically plausible indications of changes in disease activity for use in NASH drug development studies, for research use only in the US and Canada, and not for use in diagnostic procedures.
As Roger said at the top of this podcast, there will be no business section tonight. Wednesday, join Roger, Louise, Jörn, and their guests to deliver a high-insight post-mortem to ILC 2022 on surfing the NASH tsunami. Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.